Hello. Could we please get started with the next session? And also a small request for everyone. Could you please hold on to your glasses for lunchtime because we seem to be running out of them otherwise.
I don't know. Am I supposed to do my? No, I haven't. Charlie. Should I? I don't want to touch this. There's a nice man. It's fine. Fine. He's in charge. Drag it to the, the second screen that's here that you don't see. Okay, fine. That's fine. I'm very happy not to. It was mostly, I was like, I don't know how to get my presentation up. Fine, I'll go away.
Hi, everybody. Let's go back to this very, very exciting session in neuroscience. I'm very, very pleased to introduce Professor Charlie Stagg, who is Professor of Human Neurophysiology in Oxford. She's going to be talking about developing a new neuromodulation approach in a new era.
Fantastic. And thank you very much for the organizers for the invitation. It is an absolute delight to be theoretical ultrasound users. And I was wondering whether I should take the opportunity to talk you through the many, many ethical and safety hurdles we have jumped over. But I figured that, as I say, that might be cathartic for me, but will probably not actually be that useful for anyone else. So if anyone would like to know, we're very happy to talk about it. So ultrasound for us is, as I say, somewhat theoretical, but neuromodulation really isn't. And so what I'd like to do in the next sort of 20 minutes or so is talk to you a little bit about ultrasound, and there are little bits of results in there to keep your interest, but really talk about neuromodulation. And neuromodulation is something that my group and others have been doing in Oxford for a long time. And, I realize I have reached the age where, you realize you've reached an age where fashions come back round. And I'm not quite wearing flares, but I am seeing parallels, and I think many of us may be, at the beginning of this massively exciting new field to the beginnings of the last massively exciting new field. And some things went brilliantly and some things went less brilliantly with that. And I would really like us to learn from that. And I'd really like ultrasound to, make fewer of the mistakes that we made 15, 20 years ago. So we're all interested in modulating the brain. We, we're interested in that for neuroscientific reasons or for clinical reasons. And we've heard about some really fascinating clinical uses. But we're certainly not new to this. This is not something that's occurred to anybody in the last 10 or 15 years. This has been something that's been done for thousands of years, really ever since we had any idea about, that we might be able to modulate these things. So if you happen to have a headache in ancient Egypt, you would go paddling with some electric rays and get some electric shocks, and that would make your headache better. Fast forward a little bit to northern Italy in the late 18th century, and it was the first use of electrical stimulation for depression. So this was a farm worker who, if you read the case report, sounded like he had sort of catatonic depression and was hooked up to this new and exciting electricity thing. Had a few shocks and went home and said, you know what, I'm completely fine. Please, I don't need to see you again. I'm fine. In the 1920s, if you were unlucky enough to be a lady who had hysteria, who knows quite what that might be, you might have been rigged up to one of these. This is one of the first transcranial magnetic stimulation, okay, where again, people tended to be really, really happy with it and go, you know what, I'm completely fine, thank you. I don't need to do this again. We've moved on, I like to think. This is transcranial magnetic stimulation. You've heard a little bit about that this morning from Noah, and transcranial direct current stimulation, and obviously ultrasound stimulation. But history is there to sort of be laughed at and go, yeah, it's a silly lamb. What I would like to make the point of is that what I don't want to happen is for my grandchildren to be standing here going, yeah, they needed ultrasound to the brain. Okay. We need to do this properly. We need to have proper controls. We need to understand what it might be that it's doing. Okay. And I say this because some of you may be very familiar with the Geithner height curve. If you're not, then we are kind of somewhere, I don't know, sort of, that's what everybody means. Somewhere kind of here, right? We've invented it. Are we quite at the peak of inflated expectations? It sounds like a kind of Tolkien-esque travel through Middle Earth. I don't think we're quite there yet. I think we're probably about here. I have seen particularly transcranial direct current stimulation go through this. This was probably just before I started my DPhil. This was definitely my postdoctoral years. Oh my goodness, don't go there. TCS is probably kind of here-ish, I should think. I think ultrasound is probably here. I think it's a really, really exciting place to be. But if there's any way we could avoid that curve in the middle, that would be great, okay? It's not good for anybody. It's not good for our careers. It's not good for our patients. It's not good for anybody. Ultrasound is not going to cure the world. It's going to cure possibly some things. Okay. So how might we go about avoiding that? And there are lots of ways. And I think, we've heard a lot about some of the, very basics. I'm not going to labor those. We need to understand the mechanisms. And this has been a brilliant meeting. Thank you for really trying to understand that. We need to make sure we're doing things that are safe. And I think there is a feeling in the community that we really do, again, need to understand that. And I think there's a lot of willingness to share, and we must not lose that. I'd like to suggest we need to work reproducibly. Okay, it's very easy at the start of a field, especially when we've got a new shiny toy and we're all human, to think, oh, I'm going to do this really exciting thing. And we do it too. I mean, I'm completely complicit in this. Actually, it's really important to try and reproduce some other people's work too. None of us know whether our work is reproducible or not. You can't. You can just do it to the best of your ability, and you put it out there, and some of it will be reproducible, some of it won't. That's science. But we need to try, okay? And we need to be open and honest about null results. If we can't reproduce things, we need to be honest about that. And we need to have a discussion without, we'd say, well, you've managed to do this. We didn't. What have we done differently? It's not that you're right and we're wrong, or vice versa, just that there must be a difference. Something's happened. So in the spirit of that, we tried to reproduce some results. And we were new to neuromodulation. And we, all my results, I should say, in advance, all our ultrasound results that I'm going to present and talk about today are very much in collaboration with the ultrasound group at UCL, without whom we would not be doing any of this. And are led by Talika, who many of you will have seen chairing sessions here. And what we wanted to do was ask a very simple question. We're quite simple neurophysiologists. And so we first of all said, okay, let's just see what happens if we try and evoke an ultrasound potential. There's been reports in the literature that if you stimulate occipital cortex, you get an ultrasound evoked potential. Can we see one? So we tried to do as many things in the same way as the previous papers as we could. So we had a EEG setup, you can see here, that covered most of the occipital cortex. This is where the ultrasound was. We did some fairly standard ultrasound, nothing particularly exciting, and just asked the question, what could we see? We did ultrasound every 2 seconds and recorded the EEG. These are the results from O2, and in green you can see the response to sham stimulation, which was delivered here, and in blue, you can see the response to real stimulation. We cannot see ultrasound evoked potentials. If anybody can see something in that, please tell me. We really can't. Okay, and I'm not just cherry picking. I don't think if anyone can see anything in that, please tell us. There's nothing, right? So I don't know why. I don't know what we were doing differently. I think we may all have our own interpretations, but we need to explore a little bit. I think, what of these things work? Can we get ultrasound to modulate evoked potentials? Well, again, that's been well described in the field, and we've seen some data this morning about modulating one type of evoked potential. So here we looked at whether or not we could get a modulation of visual evoked potential. Again, a very objective, simple, measure of what the brain's doing. So we got people looking at a checkerboard, they hated us, and every so often we gave them some stimulation. What you can see here are just some evoked potentials. We've got the no stimulation in red and the sham stimulation in green. You can't really see one or the other, and many of you will have seen this on Talika's poster and presentation. This is not new data to you. There's really nothing there. There's a beautiful evoked potential. We can get, if you flash checkerboard someone, their visual cortex lights up. We were doing something right, but nothing. With real stimulation, we do see an effect. There's something there, I think, not clear. It's not massive. I think it's probably there, and I think it's probably significant. So here you can see areas of grey that are significant differences, corrected for multiple comparisons across the brain. And what we see, as Talika said yesterday, is this change in the very early parts of visual evoked potential. And that sort of matches what other people have seen with other types of brain stimulation, that it's something to do with the afferent volley coming in, and makes sort of sense. But it's not huge. I mean, it's doing something, I think, and I think we'd all be quite concerned if I said, well, I can change the excitability of the cortex by 20% just by doing a bit of. I'm quite pleased it's quite subtle, right? These are really robust behavior, objective measures of cortical function, and we're tweaking them a little bit, and that's probably good. But it's not the same result that's been seen before. I don't know why. There are differences. I don't know which of those are important. I would really, really like somebody else to try this, and we would be delighted to share everything we have and see what you get, and see if we can come to some kind of consensus. I think it's the only way of making sure that we can do this. So I think understanding which of our results are reproducible and which aren't is really important. I think blinding is really important, and we've heard a lot from Leonard's lab and other labs over the course of this, that the auditory confound is major, right? And it is. So I'm not going to labor this point. It has been shown before. This is our version of doing the same thing. So we did some stimulation, and we just asked, after every ultrasound burst, we said, could you hear that or not? Some of them were sham, some of them were real. Some people could hear shams, very pliant subjects. Everybody could hear the real if it's just a square wave. It's difficult to mask properly. So these were varying types of ramps, and the rest of the paper is lots of other ways that you could think about doing this. None of them are perfect. Okay. So even with really good masking and ramps and other things, that we try and improve this. It's not perfect. And you may think that doesn't matter. And I think there is a feeling, and I think there's other data here that would support the idea that this really, really, really does matter. And this doesn't matter just for really complicated cognitive things. If you're going, oh, well, I just do something very robust and physiological, and whether or not you can hear a noise is going to have no effect on that. It really is. See the posters, this matters. But to try and convince you that it matters, and to convince you that the placebo effect is real, I'm going to move out of the brain for a second and just show you this, which is my sort of favorite example of the placebo effect being incredibly powerful. So this is a Kaplan-Meier curve of a survival, okay, in a clinical trial. So this is a nasty cardiac condition, and you can see over the 36 months after they've been followed up, these are the number of people who are still alive. Okay, so this is not a kind of subtle cognitive measure of something. These are the people who are still alive. And this is a really nasty condition. So about 30% of people are still alive at the end of 3 months. In 3 years, this is a horrible condition. Now, you might think, oh, well, they're doing much better, right? Because in this group, like 60% of them are still alive. Hurrah, this is a really effective drug, right? This is all placebo, and it's matched. The difference is that people in this group thought that they weren't taking the placebo well, and in this group, they thought they were taking the placebo properly. Okay, there's no drugs, nothing else going on. They're taking exactly the same thing, and it's a placebo, but it makes that much difference to survival, right? This is not a minor, oh, well. So, you know, if you think, I guess, yeah, that is a very classic example. It's difficult to see how that can be a minor cognitive thing that we can ignore. The brain thinking that we're doing something, even if we don't tell anybody anything, is enormous, right? This is a much bigger effect than any of the drugs, by the way. The message from this very complicated study is don't worry about taking the drugs, just believe that you're taking them properly, and you'll do far better. So it really matters. We're not going to be okay saying, oh yeah, we did, you know, it was fine, we just told them to ignore it. We need to get it right, okay? And if you want an example closer to home, you know, the TMS field is littered with people for whom we didn't get this right, okay? And now it's going back and unpicking it and trying to understand what's real and what is just that effect. We need to dose accurately, okay? We need to know what we're doing. Anyone who has done this kind of thing, and it's coming from the neuroscience field, knows that these effects, you know, are small, and I've shown you the visual evoked potential results we get, right? As a neuroscientist, I'm looking at this, can we modulated something in the VEP, and there's a P of less than 0.05, hurrah, that's a P. I'm very excited. But it's tiny. And talking to our lovely engineering colleagues, they're like, but it doesn't happen in everybody, and it's not consistent, and you don't know why, and what it, okay, so it's massively variable. And one of the things we can control is the dose, and we need to, right? So I don't have data to prove this from the ultrasound, but I'm going to step into other neuromodulation for a second. So in direct current stimulation, we have shown that, you know, depending on which direction the current flows, it's either increases excitability or decreases excitability in the motor cortex. And very early on in my PhD, we looked at neurochemical changes associated with that and showed that with anodal stimulation, which seems to be facilitatory, excitatory, the motor cortex increases the ease at which information travels from the brain to the fingers, leads to a significant decrease in GABA, major inhibitory neurotransmitter. And we've replicated that, and others have replicated that. Seems a really robust finding. And actually, if you're wondering if this has a link to ultrasound, the Plymouth group have a beautiful poster showing that ultrasound to a different region of the brain leads to a decrease in GABA. So the idea that this is a way, perhaps, that these kind of neuromodulatory effects affect the cortex. But you will notice that the error bars are enormous, okay? And there's a number of reasons for that. But again, this is Talika's work. We wanted to ask the question, why? Yeah, there's lots of physiological reasons, but we don't dose TDCS. For those of you who don't use it, what we do is put 2 electrodes in the head and pass a milliamp current between the 2 milliamp current. We don't dose that on distance, you know, on current arriving in the brain. People's skulls are different thicknesses, the cortex is in a slightly different place, slightly different distance, right? So we don't take any of that into account. We just go, you're going to have a milliamp to your scalp, and we're going to assume that's a same dose. So what Talika did with in a lovely collaboration with the SimLives group is actually look using MRI and model what the current was that people received in the brain, and looked at whether that related to this neurophysiological effect that we think is important in how TDCS works. And what she saw is that, unsurprisingly, it does. If you have higher currents actually in the bit of the brain you're trying to stimulate, so if you actually do more stimulation to that bit of the brain, it responds more. And if you don't, you don't. That was modulated by slightly other things. It's a slightly complicated paper. But the basic message is that about 50% of the variance in this, it can be explained by how much current they had. Okay. So we need to do the same for ultrasound. I don't believe that's not true for ultrasound. If they're having more sonification, have bigger effects, right? Or smaller effects. So we need to really understand what we're doing, because that kind of variance is going to kill us. Okay. If we're looking for small effects and really clever cognitive effects, that kind of variance is a major problem. We were lucky to see this initially. it's quite a big effect, but the kind of things we're going after these days are small effects, and with that variant, we're not going to see it. Transparent reporting, we've seen it and it's lovely. City from Plymouth talked about pre-registering. It's great. People are pre-registering this stuff. Please do. Brain stimulation and imaging, particularly, you can do some fishing. You can find something if you look hard enough. Let's just be honest and open and say, you know what? We have a hypothesis. This is our hypothesis. We tested it. It worked. It didn't work. As a field, let's just accept that's how science should be. And yeah, go on and explore. Do some exploratory analysis. Awesome. That's where the next hypothesis comes from. But let's just be transparent about what we genuinely thought was going to happen and what was just oh, that's cool. We should follow that up. Okay. And let's publish the negative results as well. Let's not just keep all those studies in the bottom drawer because they won't be published. And I work on the editorial board of a couple of the journals. We will take negative results if it's a well-performed study and it's an interesting question. We will take the negative results just as well as we will take the positive results. The same standards apply to both. Okay, so please tell us, because this isn't going to work all the time. And if we think it is, we're just deluding ourselves. We need to power studies. You go back to TMS, and I'm guilty of this. My first studies, we were doing correlations with sort of 20, 15 people. These are small effects and they're really variable. Let's do studies with a sufficient number of people that we can actually believe the results. Okay, let's do a power calculation first and pre-register it and then do it. Open science is there to help us. I think the field has changed over the last 15 years. Please, 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 let's do it and try and get these things. And let's try and do some multi-center trials. I mean, we are more guilty than most of going after the new shiny thing. I think we should do that. That's where science is, and I think we're human. But let's also try and reproduce each other's work a little bit. Let's help each other to do that. Let's make the protocols open. Because it's not, we will get better results. The field will be better if we all share this information. And we know what reproduces and what doesn't. And what the tweaks are, right? Because there are differences in labs that you don't necessarily appreciate that might be really important. So let's try, as well as doing the new shiny cool thing, just try and do a few things across labs and see if we can come to some consensus. So I'm going to finish there. So I think ultrasound is potentially revolutionary, right? We've got this incredibly exciting thing in our hands. We can stimulate things deep for the first time, and it's amazing. We need to understand what it does. We need to understand how it does it, and we need to make it safe. Okay, and effective, right? The answer to this is not just dose so low that doesn't do anything, because then we lose the power of the technique. The answer is to work out, and that's hard, and iTrust is amazing, but trying to work out what that range is that we're happy with that might do something. And these kind of conferences are awesome. I don't need to persuade you, you're all here, but let's carry on doing them, right? The field will get bigger and it will get harder, but it is massively important to come at this from all different angles. So I'm going to finish there by thanking my research group and our funders, and most importantly, the amazing guys at UCL who pack all their stuff in a van every so often and drive it to Oxford and spend a few days, iteratively making it better. And it's been so much fun. It's proper science, it's great. And who knows, 1 year we'll actually have some results, but not yet. Thank you.
Thank you. That was really interesting. So just on the point of reproducibility. So yeah, reproducibility is of course really important, but like ultrasound is still at a stage where there's a lot of groundwork that's needed. And that groundwork needs to be laid before we can understand why results, I guess experiments aren't reproducible. So I think, it's still, we're at a good stage, so that's just one point. And the other thing is, in that first study where you showed that there was nothing, right? Nothing was going on. It reminded me of, like in epilepsy research, in order to predict, say, a seizure or something, you would look at EEGs before a seizure occurs. You would teach a machine learning, method to identify patterns that we can't see. And I'm wondering if that could be useful in ultrasound, because a lot of, what we're doing, maybe we just can't see it, but there is a difference in activity. And
Yeah.
It was just maybe speculate on that.
Yeah.
So.
No, I think those are both very valid points. I mean, I think you're right. And and don't get me wrong, I am definitely an early adopter, and we do, we have a history of doing slightly crazy things. I don't think we should take away from that at all, but I worry that if we don't, as well as doing the exciting, shiny things and exploring and pushing the boundaries, which I think we should be doing, I think we do need to make sure that the foundations are there. And that is one way of doing it. If we don't do that, we end up with this beautiful tower full of shiny things that just falls over when they pull the rug. I'm mixing my metaphors terribly. And they pull the rug underneath and suddenly the whole thing collapses, right? And I just don't want it to happen again. It's really miserable. Yeah. My postdoctoral years, when I stood and gave a half-hour talk on the wonderful things we've done, and the first question was always, but it doesn't do anything, does it? But look at all the data. Yeah, it's no fun. But yeah, so I think both have a place. Actually, yeah, I mean, I think absolutely. I'm not at all claiming that there's nothing there, and I'm hoping very much there'll be a shiny paper appearing. But we get, well, obviously there was going to be amazing things. I just think we need to, yeah, we did not confirm the hypothesis we were looking for. Might well be lots of other interesting things in there.
So when using EEG as a readout, and I'm a big fan of doing that, then what we typically need is a certain critical mass, like neurons synchronized, like a population responding. And then on the other hand, we have this highly focal simulation technique, right? So maybe sometimes a null finding might then also be due to not having the sensitivity with the recording method, because either it might be off target or just not stimulating a neural population large enough to show up on the surface as a potential, or to show up in the fMRI bolt response or something.
Yeah, no, I think you're absolutely right. And I hope I'm absolutely right. And I think there are a number of reasons that we haven't seen it and other people have. And I think that's one of them. I think there is a question always around focality. And I think as a neurostimulation field, we're always going after it. My argument about TDCS actually would be that it isn't focal, and that's good. I think there are specific questions for which focality is really important. I wonder actually whether that wasn't desperately focal. But I wonder whether that was one of them. We're actually just stimulating a larger proportion may have been the way forward. And yes, you're right. If we'd used a different modality, maybe we'd have seen it. So yeah, focality is not, you're absolutely right. Focality is not always the be all and end all.
I was curious, also given the replicability that you mentioned, what are the sample sizes that we need to use? Because what is the power that we need? What are the effect sizes that we can expect? Because if you've seen a very consistent pattern in your data, oftentimes it might have been confound and not necessarily neuromodulation. So what is your opinion on that?
I think it's a very interesting question. I think pretending that we know is probably difficult. But I think equally pretending it doesn't matter is difficult. We have powered some of our stuff from previous neuromodulation studies and sort of taken the effect size of things like TDCS and thought, well, it's a place to start, right? And what we would like to do and will do is go back and look at that effect size and tell people what it is, right? And say, this is what we found, this, and this would give you a sample required of X. And that's sort of what I mean about transparency as well. That's the kind of information that is useful, right? Because then if you were planning a study, you'd be like, oh, well, okay, I'm going to look through the previous ones and the sort of average effect sizes, whatever, and therefore I'm going to pair it to this. The field, I mean, it depends who you talk to. People have very strong opinions. I don't particularly. Somewhere between 20 and 30 seems like it's sensible number to start with, but that's picked out of thin air. I think less than that's probably a bit dodgy. More than that costs too much. Yeah, no, that's a very good point. And yeah, we try.
I'm quite convinced with the offline effects that
Yeah.
we're seeing. We have seen them for quite a long time in non-human primate, that's for sure. And now I'm convinced with some stuff we see. It's true that the online effects are a bit more difficult. So I just wanted you to comment on
Yes,
that.
no, thank you. And I mean, don't get me wrong, we're very excited about ultrasound. Maybe I should have said that in big letters at the beginning. I'm not here to get, oh, disaster. It's not. It's really, really exciting. I mean, I'm an optimist. I think there's increasing evidence from the Oxford macaque studies and from some of the human work that actually there are quite significant after effects. And those seem fairly robust, and I think they could be useful. And I think we need to understand them a bit better and optimize it a bit better. But yeah, I think don't get me wrong, after 3.5 years of ethics applications, we are going to do some of this at some point.
Any more questions?
No? Nope. Or from the one. Go on.
So, sorry, one other question. What do you think about passive sham conditions and comparing that to active stimulation conditions?
I think it needs to be done well. So I see, it's the easiest comment you see reviewers making. You send papers out to review and reviewers get hard shadows. Yeah, the control is rubbish. I'm like, yeah, but there's no perfect way of doing this. So I think a passive sham is okay if it is well matched and people genuinely cannot tell, but I want to see that data. I think you have to demonstrate it is working. I don't think you can go, oh yeah, it's fine. Like, no. If you're going to use it, I want to see that data. I think an active control is better. But then, as we heard earlier, right? Most of the things we're interested in are network-wide. It's quite difficult often to find a bit of the brain that's not involved. So I think that's not, it's very easy to go, oh, you just stimulate somewhere else. It's not easy. And the sensations and things are different as you move across the skull. So it's not, it's not an easy thing to do. So I think my inclination would be an active control if you can. And if you can't, then you need evidence that the passive is working.
Oh, thank you, Charlie. I think we're going to have to move on to the next bit. Please join me again.
Thank you.
All right, we'll have Brian Lord now talking about the default mode network and how we can modulate it with TOS.
Hi, everyone. I'm a graduate researcher at the University of Arizona under Jay Sanguinetti and John Allen. And this is my first time interacting in person with this community. And I have to say, I'm really impressed at the quality of work that's on display, the care that all of you are putting into developing this. We all agree it's really exciting, and we also all agree we have to be really, really careful about this stuff. And it's just been fantastic to see out here. So I come from Similab, and we are a meditation lab. We're interested in mindfulness and how to help people acquire more mindfulness in their lives. Let's see. Okay. So I'm going to present one empirical experiment that we did, and here's just a little overview of it. We had 29 subjects, 15 who received ultrasound, 14 who received a single blind placebo. So we knew they were getting sham. And our the subjects that received STEM, we asked them if they could hear anything, and none of them said they could hear the ultrasound that was being used. So in terms of the auditory confound, we're a little bit more confident that it wasn't as big of a deal for this because they didn't report hearing them. And I'll get into why we targeted the posterior cingulate cortex in a second. This is just the overview. And we used parameters that are supposed to be inhibitory. But I do want to say one thing that Noah talked about this morning. I'm not the biggest fan of using the inhibitory and excitatory language either. I think it's too simplistic. And we've been having this discussion in our lab, and we're also trending more towards that word disruptive, and that what we're doing is disrupting activity in some way versus inhibiting it. And we took some psychometric measures pre-post and fMRI scans pre-post. And like I said, I'll get into more of the details of this. But first, I just want to talk a little bit about mindfulness, because that's what we do at CIMA. And without getting too into the weeds, a basic definition for you is it's a certain way of paying attention to your experience. And you can break this down in different ways. You can talk about attentional control. You can talk about emotional regulation. You can talk about concentration power. You can talk about equanimity, your ability to be non-reactive to your experience. But when we talk about all these things, when we talk about mindfulness, and when we talk about why people meditate, we're ultimately talking about a type of self-regulation, an ability to sense and control your experience. And all of these different factors are all pieces of that. And the mindfulness literature has been evolving in a very positive way over the past couple decades of research. And these definitions are getting more, they're getting less fuzzy, they're getting more operationalized. But obviously, talking about subjective experience, it's always going to be difficult. It's always, you know, at that hard problem of consciousness, and but what we also think is some of the most interesting stuff in psychology. So, and also to add, mindfulness, it's kind of on this spectrum between state and trait. You can train it to be a real trait, but you can also shift into a more mindful state, such as when you meditate. Now, in terms of what drives that, the default mode network is very pivotal to mindfulness. And I think most of us are familiar with the default mode network. I'm going to talk about it a little bit. It's associated with this kind of internally directed cognition, mind wandering, thinking about the self, having kind of self-centered thoughts. And it's disruptive when you engage with the external world, but also with interventions like psychedelics or flow states or meditation. And we also know that less self-centered thought correlates with increased mindfulness and also more happiness, more wellness. And the central medial prefrontal PCC axis appears to be central to the generation of rumination. And so that's what we're interested in for ultrasound, because we thought if we can disrupt this default mode network, if we can get people out of that kind of monkey mind rumination, we might be able to support their acquisition of deeper, more mindful states. But I also want to add, we're not saying that the default mode network is bad. It's not that you want to turn this off. What we want to help people develop is more control over the default mode network. And that's what meditation and mindfulness is all about, is developing more self-control over your internal experience. So this shows how it's this central axis with the medial prefrontal cortex and the posterior cingulate that is really associated with rumination and self-talk. And we also know that meditation reduces activity in these areas. And so this is what motivated us to try to target the PCC with our ultrasound. And there's research that shows that specifically the PCC can be associated with getting caught up in one's experience, with a kind of stickiness. And in the meditation world, we associate that with equanimity. So if you are less reactive to your experience, if you don't get caught up in your thoughts and your feelings as much, that is having more equanimity with your experience. So here was our design. We took a baseline scan, and that's also where we got their structural MRI that we used for targeting. We identified the PCC anatomically and used neuronavigation to target it. Then they received either sham or real ultrasound. And then we got 2 post scans, one 5 minutes after receiving the ultrasound and one 20 minutes after, or 20 more minutes after. So 5 minutes and then 25 minutes. And then we also did pre-post with our psychometrics. So here's our system. We're using a Sonic Concepts 4-element transducer, and we used ANT neuronavigation software. Our parameters, we had a fundamental frequency of 500 kilohertz. Our pulse repetition frequency was 10 hertz. Our duty cycle was 5%. And the pattern that we used 30 seconds on, 30 seconds off for 5 minutes. So they received a total stimulation period of 2.5 minutes, but cut up into these 30 on, 30 off chunks. And so here's our connectivity results. We see this pretty dramatic reduction in connectivity using so this is seed to voxel, and we're using a PCC seed, which was our target. And we see this reduction across all the major nodes of the default mode network, immediately post sonication, and it persists for 25 minutes after. And we were pretty excited about this. And looking at the medial prefrontal, we see not quite the same full default mode network reduction in connectivity, but we do see this reduction around the PCC area. And in terms of our psychometrics, we administered the altered states of consciousness scale after they had ultrasound. And so this wasn't pre-post, this was just administered after, but compared with placebo, we saw pretty significant changes. And so people were really having, this shows that people were having noticeable effects on their experience as a result of the ultrasound. And you can see here marked, well, here's my mouse. The asterisk marks a significant effect. And the factors were merging with surroundings, the sense of time was distorted, felt a profound inner peace, and saw events from the past. The time distortions were especially some of the most significant things that people would report. Some of them reported that they thought they were in the scanner for much longer or much shorter periods of time. So it wasn't just in one direction. It seemed to just distort or disrupt their ability to track time. And it's also important to point out here that there didn't seem to be any negative experiences. There wasn't much indication of that. Most of the people that reported having an experience reported it as being fairly positive. We didn't have anyone that said it was very negative or uncomfortable, which was encouraging. And so we also administered the Toronto Mindfulness Scale because we're interested in capturing these mindfulness effects, especially capturing this equanimity effect. And there was no significant difference between the STIM and the placebo group, but we did find this interesting effect with the decentering factor of the Toronto Mindfulness Scale. And just to clarify, the decentering factor, Toronto Mindfulness Scale has 2 factors. One is curiosity, So a curiosity about your experience, and the other one is decentering. And decentering is an awareness of one's experience without identifying with it. Not getting swept up in your thoughts, an ability to kind of pull back from what's happening in your internal experience. And we plotted this against the reduction in default mode connectivity. And what we found is that as people reported more decentering pre-post, that had a correlation with their reduction in default mode connectivity, suggesting that this neural effect may have been driving this experiential subjective effect that people were reporting. And we're really excited about this because this is what we were looking for. We were looking for this kind of equanimity increase, and it looks like this type of intervention could really drive something like that. Obviously, it's we have, you know, low sample size and that sort of thing, but as an early kind of exploratory study, we thought this was pretty exciting. And it also corresponds with what other people say about the default mode network from like the psychedelic literature, that if you're able to turn some of these systems down, you can get more of these kinds of increase in well-being effects in people. So some limitations and caveats about this. We had a relatively low sample size, 29, not super small, but you know, obviously not huge either. And like I said, we didn't have a statistically significant effect in mindfulness between the STIM and the placebo group. But this is likely because, for one, the Toronto Mindfulness Scale might not be the best way to capture changes in mindfulness and equanimity. Psychometric scales can be limited. We might want to try something that's more behavioral. Not all subjects had the same response. We actually saw a wide range in functional connectivity changes across the subjects. And this is something that's really kind of mystified us in our lab, is that we see this kind of responder, non-responder split in our subjects that come in. And we think that, well, for one, all the talks this week about targeting, about modeling, about how hard it is to hit your target and know that you're hitting your target, about everything that the skull can do, all of those factors probably play into this. And we used just anatomical targeting. You know, this wasn't functional targeting. One of our ideas is to do, rather than just looking at an anatomical location, base it on the actual functional activity to target. It also needs more modeling and all the technical stuff we've been talking about today, or this whole week. So finally, our conclusions. We used these inhibitory parameters, and it looks like we got decreased DMN connectivity. And the people that had larger decrease had a larger change in their experience. We need to improve our targeting ability. A more individualized approach might improve response rate. But we think this type of modulation could be a therapeutic tool that could be used in tandem with meditation training or other therapeutic approaches. Thank you very much.
Thank you very much for the great talk. Your results have even more importance in light of recent literature, which supports pretty consistently the fact that in active, clinically relevant rumination in depressive patients, actually what is observed is a decrease in default mode network connectivity. So it will be interesting to see the translation of this into the clinical realm.
That's interesting. I'm not familiar with that research. You said it was on depressed patients, right?
That is correct.
Yeah, I mean, I've been following the DMN literature. It seems to kind of change every year or two. It gets more complicated. The things we were saying, we learn it's more complicated than that. Yeah, you know, we were basing this off of the meditation literature more than the depression literature. I would think that people with depression, there's something else going on with all of their network connectivity, right?
So like, it might seem that sort of careless, self-referential thinking, like in daydreaming, is associated with increased connectivity in the default network. Whereas during depressive rumination, actually the opposite seems to happen. But as you said, there is contradictory literature.
Well, I didn't know that. That's fascinating.
Now we're gonna have Dr. Hero, is that it? We're going to talk about offline effects, US, and cross-border.
Okay, thank you, sir. Hey, I'm Takahiro Osada from Juntendo University. So I will talk about the offline effects of TUS on human cortical and subcortical areas. Okay, so I'll talk about the response inhibition. The response inhibition is one of the executive functions that supports adaptive behavior by suppressing inappropriate behavior. In the real world situation, imagine you are crossing the street. Here. Okay, so when you notice a car coming, you need to rapidly stop actions even after the initiation of action. You stop stepping into the street. That is response inhibition. Okay, so response inhibition, from previous studies, response inhibition is known to be supported by the hyperdirect pathway connecting the prefrontal cortex and subthalamic nucleus, STN. The striatum in the indirect pathway is suggested to be involved in response inhibition. However, how the prefrontal striatal indirect pathway contributes to response inhibition is poorly understood. Okay. In the present study, to examine the essentiality of the striatum for response inhibition, we used transcranial ultrasound stimulation, TUS. This can stimulate deep brain structure. Experiments were conducted with two parts. In the first part, we tested the feasibility of TUS by measuring motor evoked potentials, MEPs, with TMS to the human primary motor cortex, M1. Yeah. In the second part, we stimulated the basal ganglia and examined the behavioral impact for stopping the response inhibition. Okay, part one. So the target location is important for TUS. So we identified the FDI, first dorsal intercells, representation in the M1 via task fMRI. The task is like this, very, very simple. Subjects were instructed to perform a finger movement task inside the MRI scanner. The subjects moved their left or right index finger by moving the FDI muscle at the rate of 2 hertz as the arrow in the screen blinked for 20 seconds each. Left movement, right movement, and the rest. This cycle was repeated 6 times during the run. Okay, so fMRI analysis revealed activation for each subject. This is a single subject result. The contrast of moving right FDI minus moving left FDI revealed activation confined to the anterior areas to the central surface. This one and this one. There is variability in the depth of FDIM1 from the cortical surface. In some subjects, the FDIM1 was located at the cortical surface. On the other hand, in other subjects, it is located deeply in the central surface. So this is the distribution of the distances. The distance based on the fMRI activation peak, the distance between the cortical surface and FDIM1 distance, and scalp, the distance between the scalp and the cortical surface distance, and the distance between the scalp and the FDIM1 distance. Okay. So for the distance between the cortical surface and the FDIM1 distance, the FDIM1 was located near to the cortical surface in most of the subjects. The median was 2.7 millimeter, and 80% of the population in our subjects was located within 6 millimeter from the cortical surface. In this way, activation site for FDIM1 was identified for each subject. Next, the detected FDIM1 was targeted by TMS and TS with the aid of an online navigator. MEPs from the right FDI muscle were measured before and after TUS. MEPs were recorded by delivering a single pulse TMS. A 40-second ultrasound pulses consisting of 30 milliseconds every 100 milliseconds was delivered to the target. This protocol was based on offline effects of a Lenner's paper. Okay. Intensity simulation by K-wave revealed that the target site was effectively stimulated. This is the MEP profiles before and after TUS. MEPs were normalized by the MEPs before TUS application. MEPs were suppressed sustainedly. And this is the average MEPs before and after TUS. These results demonstrate that TUS with our protocol induced a sustained suppression of cortical activity. Next, TUS was applied to base attack. Okay. To investigate response inhibition, subjects performed a stop signal test. The test comprised of go and stop trials. In go trial, a left or right pointing arrow was presented as a go signal, and the subjects were instructed to press the button by indicating the corresponding direction with their thumb. In stop trial, a go signal was followed by a stop signal, a pointing arrow, after a stop signal delay, SSD. And the subjects were instructed to withhold the manual response. So, not to press the button. Okay, the SSD, stop signal delay, was updated with each trial based on the performance of stop trials, allowing to maintain the stopping accuracy approximately at 50%. The test performance was evaluated by a behavioral index called stop signal reaction time, SSRT. SSRT was estimated by an integration method, and SSRT is equal approximately go reaction time minus SSD. Okay, shorter SSRT means efficient in stopping. In contrast, longer SSRT means less efficient stopping or poorer stopping performance. Okay, to investigate the striatal functions and striatal pathways, 4 steps were conducted. First, the basal ganglia was identified via fMRI during the stop signal task, and TUS was applied to the basal, identify the basal ganglia and examine that disruption of the stopping performance. And next, as a third step, the cerebrocortical region was explored as a cortical counterpart of the striatum. And next, this cortical counterpart was stimulated via TUS. Okay, in step 1, the subjects performed the stop signal task inside the MRI scanner. The contrast of stop success minus go success identified the activation in the prefrontal cortex, such as the dorsal and ventral part of the posterior inferior frontal cortex, DPIFC and VDIFC, and pre-supplementary motor area in the medial region. In the subcortical regions, the SDN, the subthalamic nucleus activation was observed, and also in the striatum, anterior part of the stamen was observed to be activated. Okay, so next, the activated site in the basal ganglia was targeted by TUS. The experimental schedule was similar to that in MAPs. 40-second TUS was applied to the target, and the task performance of the stop signal task was examined before and after TUS. We targeted the SDN and anterior stamen. As a control, posterior part of the stamen was also targeted. So SDN and anterior stamen had the activation in the fMRI, and posterior part did not have the activation during the task. Okay, this is the task performance, the SSRT before and after TUS. Sustained SSRT increase was observed after TUS to the SDN. This means the disruption of the response inhibition. And this is consistent with the DBS, deep brain stimulation to the SDN in the patient study. Also, TUS to the anterior stamen induced a sustained SSRT increase. Also, this means the disruption of response inhibition. In contrast, TUS to the posterior stamen did not show significant disruption of performance. Okay, so next we explore the anatomical connections between the basal ganglia and cerebrocortical region. We employed the diffusion MRI data from Human Connectome project. And when the seed was set to the anterior stamen, the tracks were estimated using probabilistic tractography. And these areas, mainly in the anteriorly in the prefrontal cortex, were connected with anterior stamen. So structural connectivity data, this one was thresholded with task activation. This is task activation. The anterior IFC, anterior inferior frontal cortex, AIFC, was identified as the area that had a robust structural connectivity and task activation. Next, we examine the essentiality of the AIFC, anterior IFC. Middle frontal cortex was also examined as a control. MFC, middle frontal cortex, showed that task activation, but did not show the structural connectivity with the anterior stamen. Okay, so TUS to the AIFC elicited the sustained SSRT increase. In contrast, MFC did not show disruption of performance. Okay, so summary. We applied sustained suppression effects with TUS. The anterior stamen showed the causal contribution to stopping. The AIFC, anterior IFC, is the cortical counterpart of the anterior stamen. The AFC anti-obtainment circuit in the indirect pathway works as an essential route for stopping. Okay, thank you for attention.
Thank you, Takio. So I have a question regarding the MEP study in the beginning. So you used a functional fMRI localizer to identify the representation of the FEI in the motor cortex, and used that for targeting for focused ultrasound, right? Did you also use it for targeting for TMS?
Yes,
Or
so
did you also use a classical motor hotspot search empirically?
so we did we used the online navigator for both for the TMS and TUS, and the same target was stimulated by TUS and also TMS. So we did not search with a conventional style. So just stimulated the identified the site by fMRI.
And did you then always get a proper motor evoke potential when targeting the motor cortex like that? Yeah.
Yeah. So then motor threshold by the stimulation to the identified target is very similar to the previous reports with the same apparatus. And so this means the searching method, the site by the searching method is approximately corresponds to the fMRI identified target.
Okay. And last short follow up. Sorry, it's very interesting. Because you measured the scalp cortex distance, right? So did it correlate with the thresholds? The MEP threshold?
Yes. So I will show you later.
Thank you.
Thank you so much. Just a clarifying question. What was the intensity for the MEP? And was it like before the correction for the Neurofast protocols or after?
Yeah, actually, before the correction of the Neurofast, and we use 500 kilohertz. So the intensity is a little bit. The actual intensity is a little bit larger than we expected. So by the factor about the 2.8 or so. So then, for the FDI MEP results, so the intense ISPPA is about 80 or so, a bit larger than conventional style. Good.
Thanks. Very interesting work. I also have a question regarding the MEP amplitudes where you show this very sustained suppression. Did you do any follow-ups, any additional analysis to see how long-lasting this suppression actually was?
Yeah, so actually we did, so to be honest, we did not systematically investigated the time course, time follow-up of the time courses after the 60 millisecond application. But in some subjects, the MEPs were recovered at the approximately at 90 or 120 minutes after the TUS application. But the average data was not obtained in our research. Thank you. Hello, everyone. The title of my presentation is about the effects of different sonication parameters of several tasks of human motor cortex. I'm a postdoc fellow in Dr. Bertrand's lab at Cranberg Research Institution. A typical brain stimulation include the DBS, TDCS, TMS, and ultrasound. The spatial resolution of the the spatial resolution of parts can be 1 to 5 millimeter, and the depth of simulation can be 15 centimeter. And these two features very close to the DBS and superior to the other two non-invasive brain simulation, TMS and TDCS. The current non-invasive brain simulation protocols include the TMS and the TDCS. The TMS protocol includes 1 Hz RTMS, regular TMS, and 10 Hz regular TMS, and the continuous burst TMS, and the intermittent burst TMS. The TDCS has a load simulation and a Kessler simulation. Some protocols have inhibitory effect, and some protocols have excitatory effect. The after effect of the current non-invasive brain stimulation protocols are determined by the TMS initiate motor-evoked potentials. If the post-MP increase, the outer effect is excitatory, and if the post-MP decrease, the outer effect is inhibitory. Our previous work proposed the Stepper task. The Stepper task was defined as an 8-second train or 20 millisecond ultrasound burst repeat at 5 Hz. And the time burst duration is 20 milliseconds. The PF is 5 Hz, and the sonication duration is 8 seconds. And we also design a regular task. The regular task has the same total sonication duration and the prototype of the Stepper task. And this is the MP result. The top panel is for the average MP. The bottom 3 panels is for individual MP. The red line is for Stepper task, and the blue line is for regular task. The back line is for sham Stepper task. And we found the Stepper task can increase MP at T5 and T30, but the regular task and the sham task have no effect on the MEP. So we think the 8-second train of Stepper task enhance motor cortical excitability for more than 30 minutes. Now this work is intended to explore the effects of different sonication parameters of Stepper task on human motor cortex. 4 sonication parameters were studied: acoustic intensity, sonication duration, PRF, and the duty cycle. Acoustic intensity was tested with 10 watt and 20 volt. The sonication duration was tested with 40 seconds, 80 seconds, and 120 seconds. The PRF was tested with 2 Hz, 5 Hz, and 10 Hz. The duty cycle was tested with 5%, 10%, and 15%. In this study, 14 health subjects attended 8 visits on separate days. One visit applied the standard Stepper task. The parameter of the standard server task, just the board parameters. And in the other seven in the other 7 visits, one solicitation parameters was changed. The TMS measures were tested before, and the 0 means, 30 means, 60 means, and let means after the server task. The TMS measures includes single path and the per path. For per path TMS, there are two stimulus. The one is condition stimulus. The other one is test stimulus. If the condition stimulus is 2 milliseconds before the test stimulus, the MP decrease, and we call it as short interval intracortical inhibition. That if the condition stimulus is 10 milliseconds before the test stimulus, the MP increase, and we call it as intracortical facilitation. But if the condition stimulus is a few milliseconds after the test stimulus, the MP will increase, and it is called a short interval intracortical facilitation. And the 3 intervals were tested in this study: 1.4 milliseconds, 2.2 milliseconds, and 2.8 milliseconds. This page is a result of acoustic intensity. The left panel is for the single path TMS. The red panel is for per path TMS. The figure A is for the average MP. The figure B and the figure C is for the individual MP. In figure A, the backline is for 20-volt server task. The blue line is for 10-volt server task. And all the post MP are normalized to the baseline MEP. So if the value is higher than 1, it means the effect is excitatory. The field marker means significant relative to the baseline. The empty marker means no significant difference than the baseline. So we can find that the 20-volt server task increase the MP at T0, T30, and T60. But the 10-volt server task have low effect on the MEP. In the right panel, the top row is for 20-volt server task. The bottom row is for 10-volt server task. For SICI, the value is lower than 1. And so the more the value close to 1, the less the inhibition. For ICF and the SICF, the value is higher than 1. So the higher the value means the strong or the more the excitation. So we found the 20-volt server task decrease the SICI and increase the SSF, but didn't change ICF. And the 10-volt server task have low effect on all the intracortical cycles. Okay, this page is for the result of PRF. Same as the previous page, the left panel for single path TMS and the right panel for per path TMS. In figure A, we can say the backlines for 5 Hz server task. The red line for 10 Hz server task. The blue line is for 2 Hz server task. We can find the 2 Hz server task produce changes just after the stimulation, and the 10 Hz server task increase the MP for more than 30 minutes. But the 5 Hz server task can increase MP for more than 60 minutes. And this is the individual MEP. The red panel is for, okay, the top row is for 2 Hz server task, and the bottom row is for 10 Hz server task. We can find the 2 Hz server task have low effect on the intracortical cycles, but the 10 Hz server task also reduce the SICI and enhance the SSF and didn't change ICF. Okay, this page is for the duty cycle. In the figure A is the RG MP result.
The red line for cerebral task with the 15 duty cycle, the black lines for cerebral task with 10% duty cycle, and the blue line is for cerebral task with 5% duty cycle. We can say the cerebral task with 5% duty cycle can increase the MP for 30 minutes, but the cerebral task with 10%, 50% duty cycle can increase the MP for more than 60 minutes and 90 minutes. In the red panel, the top row is for 5% cerebral task, and the bottom row is for cerebral task with 15 duty cycle. And we can find the cerebral task with both 5% and 15% and decrease the SSF and increase the SSF, but didn't change the SF. And this page is for the result of sonication duration. We can look at the figure A is also for the average MEP. The red line for 120 seconds simulation, the black line for 80 seconds cerebral task, and the blue line for 40 seconds cerebral task. We can find 40 seconds cerebral task can increase the MEP for 30 more minutes, but the 80 seconds and the 120 seconds can prolong the effect to 60 minutes and 90 minutes. And in the red panel, the top row is for 40 seconds simulation, and the bottom row is for 120 seconds simulation. We can find that both 40 seconds and the 120 seconds simulation, cerebral simulation can decrease the SSI and increase SF, but has no effect on the SF. Okay, so at first, the acoustic intensity should be 20 volts to touch the home cortex with our ultrasound device. And the corresponding ISPPA can offer reference for other device. The ISPPA for our for this for 20 volt cerebral task will be 2.3. Yeah. The optimal PRF is 5 Hz for cerebral task to induce homeoplasticity. And cerebral stimulation actually mimics the pattern neurofiring in rat, and the combination of a complex spike pattern gamma frequency with the theta frequency repetition rate. And such pattern is also known as theta gamma with amplitude coupling. Higher duty cycle and longer sonication are associated with longer duration or increased cortical excitability induced by cerebral task. And the non-sonication duration could be a better way to prolong the effect. Some subjects feel strong sensation with high duty cycle. For example, I think at least 4 of my subjects can feel strong sensation for the cerebral task with 15 duty cycle. We also can find all the cerebral tasks with different parameters can enhance the motor cortical excitability, but all of them reduce the short interval intracortical inhibition and enhance short interval intracortical facilitation, but had no effect on intracortical facilitation. And the effect of cerebral task on cortical excitability may be in part mediated by changes in the intracortical circuits. Okay, thank you for your attention.
Thank you. Just curious, what do you think about the possibility of the rhythm entrainment by the acoustic or tactile sensations from the stimulation? So we know, for example, if you present visual stimuli with certain frequency, then you will have entrainment also in the brain rhythm. Do you think something similar can be happening when you are using tooth here? So in vision science, we know that if you present visual stimuli with certain frequency for repeated times, then you will get entrainment that is also evident in the EEG, for example, or MEG. And here you're also presenting stimuli, acoustic or tactile, with a certain frequency for prolonged time. So do you think some kind of entrainment can happen due to the, well, acoustic or tactile confounds?
Yeah, I think so, because yesterday, Chinese researcher also report exciting effect with PRF of 2000 hertz, but get inhibition effect with PRF 50 hertz.
So did I understand correctly that you did like change one of the parameters at a time, right? So either duty cycle or duration, right?
Yeah,
In different experiments. Did you also then combine always the best working parameter from each dimension, basically, and do a 120 seconds 15% duty cycle, 20 watt. So taking always the best parameter and combine that?
Yeah, I think the best parameter should be at first the PRF should be 5 hertz, and the transmission duration should be 120 seconds. Yeah.
But did you ever run a paradigm where you actually took the best parameters from each of the experiments and combined them all, like the best parameter the best protocol you could create, basically based on your results?
Okay. Yeah. For the best performance, I just compare the effect among the 88 experiments. Yeah.
You know, to talk later. Thank you.
Also. Thank you. Thank you.
We're slightly delayed, so I would like to have lunch now. That's okay with everybody? Okay, good.